Right. So welcome everybody to this seminar of the IBM series. Today we host, with a bit of conflict of interest, Thomas Schmidt, which is also part of the organizing committee of the IBM. So I'm going through his CV real quick. So he's German, but he studied in California, and in particular he got his PhD in studying NMR, biochemistry NMR, at the University of Southern California. And then he moved right away to the NIH, the National Institute of Health in Washington, as a visiting fellow or so postdoc, I guess. And then he was promoted to scientist there in 2021. Among the awards as a student and postdoc, I cite the John Weil Young Investigator Award from the International EPR Society, and that was in 2021. And yeah, now Thomas is still at NIH as a scientist, and today he's going to talk about deer data analysis, and because he's an expert on the methodology. So Thomas, you're welcome to share your screen.
Sounds good. Thank you very much, Thomas. I know there are so many Thomases here. All right. Okay. Well, thank you everybody for attending the seminar. Before I do start, I wanted to make a little announcement. Max Tesmer and Stefan Storlab, they're going to have a workshop and coming up on May 22nd. And it's around a 3-hour workshop. It pretty much treats the idea of how to paramagnetically label a protein and how to utilize those pretty much models to extract distances information, docking information, and other things. So I highly recommend it to visit Kai Live. If you did not receive the email, I'm pretty sure they haven't posted. Otherwise, here's a little link posted on the slide. Please go ahead and fill up the form and you're ready to register. Now, as Thomas said, I will be talking today about global analysis of deer data. Now, that being said, why is this important? Well, it is important because normally processes, like when we measure deer spectroscopy, we obviously get biomolecular information, such as distances, and we always care about conformational exchange, how the structural arrangement is, but that does not really give us insights of how actually the protein is using that structure to fulfill a biochemical function or biological function. And in the end of the day, this is really what we care about. We don't care necessarily about the distance by itself, but we care about how we can utilize the distance to explain this biological function. Now, before I actually continue, I know I'm going to repeat myself, but I probably need to go quickly over what is the deer spectroscopy or what is a deer experiment. Now, this deer experiment is very simple. It really just cares about distance measurements. That's what it really does. And normally how we apply the deer experiment is that we have a question. And the question normally proceeds that we have a protein, let's say, for example, with 2 domains. In this case, it will be a red and a blue domain. Now, our question is, well, how are those 2 domains arranged in the 3-dimensional space? Of course, like if I just fulfill that domain, I put it in AlphaFold or any other software, I'll probably fold it, but I probably won't know the arrangements. So therefore, that is an open question, and simply put, one distance is to solve it all. And therefore, we would take our protein and we would attach conveniently attach a paramagnetic label, such as our favorite MTSL, to free cysteine residues. Upon covalent linkage, those paramagnetic residues are now arranged in space. And if they're arranged in space, they are also dipolar coupled with each other, meaning that we now can perform a deer experiment and observe this coupling. And this is visualized through this trace, dipolar evolution time trace. And as we can see, there is in red, for example, we have a dipolar oscillation, a waving function that subsequently, after mathematical manipulation, now can be translated from the time domain, from the dipolar evolution time domain, to a distance domain. And in this distance domain, we need to keep in mind that it's actually a distribution, what we're observing. That distribution can now feed back into my protein. So we started with a random arrangement, but upon feeding that distance back into my protein, I can now restrain that conformation. And upon restraining that conformation, I can now ideally have a representation of how those two domains are arranged in space. Now, in my hands, normally we use different methods to it, and obviously we use isotope labeling. Now that's all good, but one thing what I'm going to talk today about is more the idea of global analysis. And now there are different ways of analyzing the data, right? So there are different mathematical tools available to go from the time domain to the distance domain, from the dipole evolution time domain to my PR distribution. And of course, there are many packages available. Just to cite some of them, of course, there's that the SVD package available, developed in the Fried lab and further developed in Madurska-Vastava lab, and very famous one, Stefan Stoll and Gunnar Jeschke, is always the DIA-analysis and subsequently the DIA lab software packages that really make it available. However, the one thing what I did become fond of, and obviously there's nothing wrong with the other ones, I'm just kind of rather fond of it, is the global analysis as performed with Gaussian fitting, so paramagnetic fitting, which was pioneered by Eric Husted. And the idea, what I like about it, why I really dabble into it, is that I can actually describe a mathematical process that underlines an array of dipole evolution time traces. So in this case, I really want to make it clear that we don't really care necessarily of the distance distribution, because we're really just fitting the dipolar time tracer to a parametric model. Now, this is what we're going to treat about. Now, furthermore, we also need to very quickly keep in mind when we fit, what does it really mean? So again, we would have most likely a dipolar time trace present, right? And now we're going to fit a model to it. Now the model selection is very critical, right? So if I select a model, so for example, here we have a dotted line that presents a curve. However, if I fit a linear curve to it, that is a very poor fit. And it can be pretty much easily identified as a very high chi-square, reduced chi-square value. If I now go the next step and I actually find the right correct mathematical model, actually my reduced chi-square is equal to 1. That means that indeed my mathematical model describes the underlying process that occurs in the data. However, there's another opportunity, and this is a very risky thing, is that I overinterpret the data. In this case scenario, now I'm fitting even more involved mathematical model that fits all the points. Now that I pretty much have noiseless data. But in this case, I'm actually working with an overfitted model. The model is overfit. That means I'm overinterpreting the model. It's not. It takes not into account the noise, and that can be easily identified as reduced chi-square is less than 1. So this is normally what one can think about. That when I'm talking about model fitting is how well can I mathematically describe a biomolecular process that can be described by the data without overinterpreting the data, without thinking that noise becomes signal. In my hands, I normally like to try to use global analysis software in Python called LMFit. I'm very fond of it. It's very easy to use. Again, it's coding basis. And it just allows a variety. It really allows you to build your own model, to restrain global, local parameter, different. It allows you to do different optimization function. And what I would do in the subsequent slides from here, I really want to show how to use this program so that everybody in the community is actually able to pretty much utilize global fitting for their own purposes. Now, given initially I would not go right away into DR, I would just stay with a simple Gaussian fits, and the architecture of the software or architecture of the Python code, which I normally utilize, and we will go from there. Let's see how that goes. So how do I set up a global fit? Now, that does not mean necessarily in this case it's just simply a Gaussian. You don't have to necessarily directly follow it. This is normally how I set it up. I normally describe initially the function. This is the mathematical expression that I want to fit to my data. So if I, for example, assume that I have a Gaussian distribution of data points, and I want to fit a Gaussian model to it, well, then it will be a Gaussian function as shown here, right? Now, the next thing is I need to have my data set. So each data set which I feed in, now this could be infinite amount of data sets, but let's say I have one data set presenting at a signal to noise of 0.3 and the other data set is 0.5 and on and on. Now, all of them describing the same distribution, the same process, and therefore it should be fed in directly. So here in this case, we have a Gaussian data set that will be fed in, and it can be described simply by amplitude, the center function, and the width of height for the Gaussian. So meaning that X is a number of data sets I have, and then we have the parameters, which is amplitude, center of the function, and distribution. So all of those three can now describe the Gaussian very easily. Now, at last, so what do we have? Right now, we have a mathematical expression. We have a set of parameters for each data set. And at last, now we need to have our objective function. Now, this is most important, actually, to be honest. And the reason why it's most important is, how do I know that I have a good fit or a bad fit? In my case, I normally just use the residuals, minimize residuals. Here we go. This is for my end, this is normally working the best. But you can, of course, decide what other expression you want. So how do you decide that you have overfit, underfit, or perfect fit? In my case, I just take data minus my model. So meaning that I have my data set minus my model giving me the residuals. And if it's a perfect fit, in this case, I have a normalized distribution of the residuals, and I'm good to go. So this is normally how I set up my global analysis scripts. So what do I get out of it? If I do a global analysis, what do I get out of it? So in this case, I have now multiple data sets. So all of them have different Gaussians. They're all around the same width and different height. But all of them have actually the same width. They have different amplitudes, they have different location, but all of them the same width. Now, doing that, I can now actually describe this. I can set up my parameters for my subsequent fit by saying, okay, I have an amplitude of my Gaussian. Okay, this is my starting condition. And for all of them, I'm going to set up some random amplitude and some random center of the location and then my random distribution. But my width, I'm going to say that the width for all of the Gaussians, and this is in the second part, for all of the Gaussians, the width is going to be the same, actually. So what do I mean? It means that while all the variables for all those 5 different data sets can be random, the one thing which has to be the same for all of the data set is my width. Okay? So if I fit the data subsequently by just simply minimizing my residuals, I can now extract my parameters, I can restrain the fit. Now, if you look at the first figure, you can see that actually the data is very noiseless. It's very clean data, almost no noise, no problems. Of course, I can fit it. There's no issue. I can even fit it if I don't have a global parameter or shared parameter. And this is true. But if I now go in subsequent slides, in the subsequent figures, you will see that I pumped up the noise level. This noise level becomes so bad that for certain, I would not be able to actually characterize those Gaussians anymore, just because I have too much noise. However, upon restraining the distribution and saying that I have a shared parameter between them, I actually can still fit them relatively well, even with a large number of noise, simply by just saying that one parameter is in common and can be restrained. I hope that makes sense, because this is really the idea of global fitting, that I can take a parameter which is in common between multiple data sets. And if I can now say that that parameter is restrained to the data set, I can actually extract those information quite easily. Now, this can be also done, of course, not just with one Gaussian, but let's say with two Gaussian. In this case, very simply, I have a bi-Gaussian distribution at different noise level. Same idea. I have two width, I have two amplitudes. And I'm going to look at multiple traces. Now, what you're going to see initially, if I fit one single Gaussian, it doesn't fit. So I have to add another Gaussian to really show that there's a bi-exponential fit. So again, this has to do now that the distribution can change, again, by just sharing a parameter. If I add noise, of course, if I don't have a global parameter, don't restrain my parameters, I can't fit it very well. But by restraining it, I really lever that restrained fit, and I actually can still extract data, which otherwise would be quite impossible. Now, okay, here we come more to the interesting part. So how do I go about it if I have a Deer curve, right? So if I have a Deer curve, a dipolar evolution time trace, and I want to extract a single Gaussian out of it. Now, this is not a big deal because I can translate it over. I can translate over as described in the global analysis by Eric Husted by simply calculating what would be the distribution and then back calculate what is the expected time domain function. Now, if I have two Gaussian, for example, I now need to separate them out and they need to be normalized, meaning that the integral of both of those functions need to be 1. And then again, I can just translate it over to a dipolar evolution time trace. Now, however, what I want you guys to pay attention right now to is that both of those Gaussians have a center and a width, right? And obviously an amplitude. And the center and the width of them can be easily restrained. Those are my parameters which I restrain. Now, again, I want to go over to the Python script. If I look at the bottom, calculate distribution, you will see that all those variables come into play. Center 1, center 2, distribution 1, distribution 2. Those are parameters which can now subsequently be fit. Those are fitting parameters that I can actually restrain. So this is actually the connection how I can utilize the software packages or the Python packages by simply defining what parameter is in common between different data sets and which ones I want to fit subsequently. So let's go one step further. So let's talk about what's the difference if I have a Deer trace, I have a Deer time domain. What are the differences between the local and a global parameter? What are parameters that I can keep global and which ones I have to keep local? So in my experience, normally when I have a time domain trace, it has an amplitude, obviously. Normally we normalize it to 1 to just make it visually appealing. But this is pretty much a scaling factor. That scaling factor will be always offset. And so every time trace will have some scaling factor. And furthermore, the modulation depth. The modulation depth in general has a slight variation. So my experience is it's very good to keep that as a local parameter. You can do a global parameter out of it, but it's very easier to understand if you keep it a local. So because those two parameters, they actually have to do with a time domain, and they don't really feed into our distance distributions. So that's kind of easier. Now, In regards to our two Gaussians, so let's again, so what is our global parameter? I already said we have a Gaussian. The Gaussian has the center and the width and the amplitude of the Gaussian, right? Normally the center and the width, they can be as a global parameter, because if you have multiple data sets, then that will probably change the amplitude. But if it's the same protein, most likely the distance and the width doesn't change. And that holds true if you have two Gaussian distribution as well. We simply now have actually, well, two width and two centers, right? So they will always be the same between all data sets. And at last, I want to propose something else. Well, as I mentioned, if I have a protein and I, for example, add and I add a ligand and the populations are changing, but the center are actually staying the same. So you can see that very nicely if I just might simulate a data set where I have a distance at around 40 angstrom and a distance around 30 angstrom. Now, as I, for example, add my hypothetical ligand to it, you can see that one population actually decreases severely to like close to zero, or absolutely to zero, and the other population increases all the way up to 100. So that means, however, what you can see is that indeed the center of the Gaussian and the width stays the same, but only the amplitude changes. But more interestingly, think about one step further. Well, what if actually the amplitude change can model a biological function underneath it? Well, this is great, because if this is true, then I can actually model the binding event or a physical function like a relaxation based on the decrease or increase of this amplitude. So those ones becomes now a restrained global parameter. So initially we had the amplitude as a local parameter, but upon deciding that indeed the increase and decrease is a function, let's say, of relaxation, I can now model it as an exponential where I simply have an exponent as my global parameter. Now, this is important because we are now, so more global parameters we have, so better we can restrain the model, right? So it's more we can actually restrain the underlying function. Now, what I propose to you today, what I described today, is pretty much the global analysis, just like step for step, what I normally go about. But of course, I should also show it on how I go about an example. So normally what I do, I would have taken my own data quite elaborately. But today, I think it would be nice if we one time just look at some previous data, which I think could have been done better of my own, where I actually modeled it without global fitting. And now I'm looking at this data from many years ago and look at it through a global analysis looking glass. And subsequently, we want to look at some very beautiful data, amazing data set collected by Tobias Hutt, and how global analysis can actually help to maybe tease out a little bit more on this data set. All right. So the first step, what we're going to look at how we can apply global analysis is in a relaxation mechanism. In this case, we're looking at something called a T2 edited Deer experiment. The T2 edited Deer experiment itself, what it does, it leverages isotope labeling, meaning if I have a protein, in this case is protein A will have two distances, the A distance and the B distance. Now, however, if everything is deuterated, the distance will be the same. However, upon exposing one of the paramagnetic labels to a localized concentration of protons compared to deuterium, that distance will actually decay as a form of relaxation. Because of the coupling between the electron and the proton, the electron relaxation decays, or the T2 relaxation decays faster. And that was already well established by David Norman for a while. Now, where do we go with that? Or what I want to say, what I want you guys to take away from here is that if I have a T2, like if I have a protein, I have two distances, A distance and B distance, which are due to row number distribution. And that can be shown here on the distances. If I now introduce localized protonation, one of the distances will actually dramatically decrease in comparison to the other. And upon doing so, one can now extract the information like the T2 relaxation and probably isolate information on the localized environment. However, back then when I did this study, I actually only simply just used, back then it was still de-analysis. And I didn't do any parameterized fitting. I did de-analysis in SVD and then subsequently fit Gaussians to the distribution to extract the population. Now, however, what I'm proposing now is, well, instead of doing this procedure, why not go directly to the time domain? So now we're going to apply what we have learned in the previous slides on global fitting, going to see how we can apply it. So if I have my dear data set, I'm going to fit now to the time domain my Gaussian distribution. In this case, we have a 3 Gaussian distribution, one small blip, and we have a distribution at around 31 angstrom and around 38 angstrom. If I now assume that each Gaussian can do whatever they don't have any global restraints whatsoever. They just have 3 Gaussian. I just fit it directly to the time domain, let the amplitude, let the width, let the center do whatever they want. If I do that, I get the gray distribution. You can see it's very noisy because the data itself was not perfect. Now, however, if I now say, well, hold on there, let's restrain the Gaussians. Let's say I have a center of the Gaussian which always stays the same. And let's say I have a distribution of the Gaussian which always stays the same for all the different data sets. So if I do that now, and one of them is decreasing, the other one is increasing, then I actually can get more information. I can get rather good information on the amplitude, because the amplitude is the only localized parameter from the fit now for a Gaussian towards dipolar evolution time traces. And you can see very nicely that is compared to the gray one, the global fit is rather smooth, right? But is it as good? Can we do better? This is my question now. In this case, I have, if you can see on the bottom of the right side, we have 6 parameters which are global, right? But we still have the amplitudes which are local. Now, what can we do? And if I say to you already, I already said that actually the Gaussians itself would decrease as a function of relaxation. You right away would think, well, hell of it sounds like an exponential somewhere. And that's correct. In this case now, we can actually take the deer data set and restrain the amplitudes of the Gaussian to an exponential, right? So then now when we do that, we go from what we had before, in which case we let the amplitude be a local parameter. We now fix it to a rate constant. So they're all the same rate constant, and we're going to observe as a Gaussian increase or decrease as a function of that global parameter, that global exponential function. And indeed, if we do that, we can now get a very relatively good, smooth, gray Gaussian, and it looks very nice. Now, however, the truth of the fit is always in the residuals. This is my experience. When I do the fit now, if I do the fit of my parametric fit, there's a lot of variables and there are a lot of methods to actually quantify of how good it fits. Whatever number comes out of it, I just urge everybody, always look at the residual, because sometimes you think you have a good fit, but you see there is some systematic error that cannot sometimes not be very well evaluated by whatever test you're using. And so I really urge you, please look at the residuals at all times. So in this case, of course, the residuals look perfect. I've reduced chi-square of pretty much 1. That means it's a close to perfect fit, and that's nice. Now, let's look at something else. We are again treating the same idea, the same model. We have our protein A. Again, we're looking at relaxation. In this case, we're looking at relaxation not of an MTSL label, but we're looking at the distance distribution as a function of relaxation of an R1P label, which got famous through Wayne Hubble. Now, the R1P label is a little bit different from MTSL label because it has that ring pyrene ring on the side that makes it more global and therefore restrains its conformation. Ultimately, what we get is we get sharper distances because of that. But nonetheless, we still have 2 distances which are rising from the 2 rotamer states. Also, again, we introduce a localized proton moiety, in this case a leucine. And what you're going to see now is very interesting, because what I'm going to describe is pretty much how I would work through this problem and try to maximize my fit. All right, so initially, again, we take several Deer traces as a function of evolution time because the relaxation process. And what we're going to see is that, again, we have 2 Gaussian distribution. In this case, it's just 2 Gaussians. And if I do not restrain the center of the Gaussian or the width of the Gaussian, I have obviously my Gaussian distribution. But as the noise gets more and more, we can see that in the gray, the Gaussians, the gray ratios, you can see that it varies quite a lot. Now, in the next step, I'm going to now restrain my mean distance, my mean distribution for those 2 Gaussians. So you can see that all of the Gaussians now the same center and the same width of it. Now, as I go on now, you can see that that now actually my function, my ratio between those 2 Gaussians becomes rather smooth compared to what it was before. But again, we should do better, right? Because we already announced to you that's a relaxation process. And if I say relaxation, of course, I'm going to think about exponential. So now, instead of letting the amplitude go local or freely, I'm going to restrain the amplitude. And I'm going to restrain the amplitude to the exponential, and therefore pretty much as one exponent. So it becomes a global variable. So I go from local to global, and I can restrain it more. In this case, if I do that now, again, I get that gray line. The gray line represents my exponential fit. But if I look at the amplitude, I mean, you can look at that at the time domain, but even if I look here at the distance domain, if I look at the gray line, which is when I restrained my exponentials and my distribution, you can see there's a waving function up and down for the amplitudes. And that kind of means that it's not perfectly fit yet, because I didn't do good enough yet. There is some function that underlines that I was unable to fit. So in the next step, I'm actually adding one more function to it. So in this case, I'm now keeping my mean distance and my distribution for the Gaussian fixed. So those are my global parameters. I have my exponent as a global parameter. But then in addition to it, I'm also adding an electron-proton coupling distance, so a dipole interaction with electron and protons as a function of relaxation. So now if I do that, you can see that now I'm obtaining a gray waving function that perfectly describes the underlying data set. So how do I go about it? So again, this becomes very centered. So again, I now need to feed in all my information, my mathematical model. So initially, I'm going to start up with describing an electron-proton coupling. So this is like the distance between the electron and the localized proton moiety. Then I obviously need to normalize my distances just to get it to amplitude to 1. And I can now translate from time domain to distance domain, as you can see. And at last, again, I have my objective function. So what is the last step? In the last step, I need to describe my parameters. Those are the parameters which I'm actually going to fit, okay? So here is a number of parameters where we have like all the parameters described, which is the relaxation component, the center of the Gaussian, the width of the Gaussian, the amplitudes, the phi angles, all those parameters can be perfectly described in just a simple mathematical description. And then at last, I now want to set up my global parameters. This is the most important thing. This tells me that my entire fit can be parameterized as with only a very, very few local parameters. So the more global parameters I have, so more I can restrain my fit. And at last, as I mentioned before, I can now fit my time domain shown in red. And red is the actual fit, and blue is the data. And obviously, I need to look at my residuals as an output. Again, the residuals look relatively good, not as good as before. And they have a reduced chi-square of 1.15. So I did fit the data very nicely. I do believe I can do better, probably with just playing around with the data. But you can see that in the process of iterating through different models and different mathematical expression, I can get a better and better hold on my data set. Now, at last, I don't think we have time for the bonus, but at last, I just want to touch on a project based on actually Tobias Hetzel and Olaf Schiemans, who recorded the data set a while ago and published like a landmark paper on rapid freeze quench. So pretty much the idea is to actually look now at proteins, because obviously that's what we're interested in about. And we're going from the apo state to the hollow state, where we change in conformation due to the binding of cyclic AMP. And that can be easily expressed again if I think about in a dipolar time domain, that one dipolar time domain overcomes the other one. And so this is exactly what Tobias Hetzel and Olaf Schiemans did. In this case, they, of course, used their amazing rapid freeze quench rapid freeze quench machine, and pretty much mixed their protein, the ion channel, with cyclic AMP, and looked at the binding event itself. Upon doing so, now they have dipolar evolution time tracer at different time intervals. And what you can see if you follow it, if you just do a simple D-analysis or D-lab Tikhanev regularization, you can see that one population increases while the other one decreases. And this is very fascinating. This is very great, very well done. And then what both of them did, they fitted the data to a beautiful binding curve and could extract information on the welling time, like how a ligand binds, how the conformation change occurs, in which case the free protein would take some time, where it has a dwell time before it actually produces the conformation exchange upon binding. So the ligand literally just sits in the pocket. Now, however, so this study was actually done very, very great. But what I did notice that there was some little deviation. So actually, me and Tobias, we chit-chatted about it during one of those IVM talks. And I asked him, and he was very nice to actually share the data. And so, yeah, share the data with me. So that was very nice of him. Upon receiving the data now, of course, the first step is to identify what distances are underlying in this distance distribution to reproduce what Tobias actually has done. And so simply we utilized CalcPR, which is a program in the NIH, Explore NIH software package, to remodel those distances. You can see we can do very well by simply just calculating those distances, simply just on crystal structure, so we can reproduce it. That's one step. So if we can reproduce the distances, then we can obviously fit Gaussians to it. In this case, that's what we did. And indeed, simply taking the data, we can produce the Gaussians. Now, the apo state, which is the black one, can be beautifully reproduced by simply just 3 Gaussians, as shown here, a 39 angstrom distance, 34, and a 40 angstrom distance. And they should be, in theory, all under equilibrium with each other. So now we're going from a global fit, we're looking at now fitting different rates and exponentials. So we are looking at differential equation fitting, okay? So we look at kinetics. Now, when the change is occurring though, so now we're going to look at the other one when it actually has gone to the fully hollow state. And again, the hollow state can be characterized by two Gaussian that seem to be in exchange with each other. So we have the hollow state and the apostate. The hollow state has two Gaussian, they're under equilibrium with each other, therefore have two distributions. Now, since we identified the Gaussians that are included in the apostate and the Gaussians that are included in the hollow state, we can now simply just extract their population just simply based on a Gaussian fit. We did not restrain the amplitudes yet. This is just simply restraining the width and the mean distance of individual Gaussian. You can see in the subsequent figures that some Gaussians will go up, some will down, some will increase. And they're actually all over the place. But more interesting is that the Gaussians don't all increase or decrease with the same rate. They're actually increasing or decreasing differently. This is very important. They're increasing and decreasing differently because they have different propensities. Now, what do I mean? So if everything would be perfect, if all of them would be under equilibrium and I have my apostate just going over to my hollow state, just directly, then all of those Gaussians would actually disappear equally. All of them do. But that's not the case, actually. They go down at different rates. That means the specific labels, specific parametric labels, and a specific location have a preference to go through a specific pathway before they orient themselves. So they try to orient themselves before they actually go over to the hollow state. And this is exactly what we see. So if everything would be under equilibrium, as we shown here, the diagram, what would happen is that we have a rapid decrease and an increase, and it would go to an equilibrium, would decrease at an equilibrium rate. But that's not the case. We actually, if we follow the Gaussian, you can see that the blue one, they actually decrease to zero, meaning that there's preference to it. And therefore, we actually had to remodel the idea where we have one Gaussian to the 39 angstrom going to the 40 angstrom, but it does not exchange with the 34 angstrom. So there's a distinct pathway that those MTSLs will take before they actually go over to their hollow state that is under equilibrium. And this is very important, and we can actually describe it very nicely, this equilibrium function, in which case we now see that we do fit all of Tobias's DO data very nicely. You can see the residuals very clearly. It's very well done. And the beautiful thing on this data set is, which is amazing, that everything was recorded in triplicates. So when I fit it, I don't just fit it in one row. I actually fit each trace 3 times because it's done in triplicates. So it's producing very, very accurate measurements. So at last, we can now then identify, based on the global fitting, that indeed we can go from the apostate to the hollow state by rearranging those kinetic pathways where one Gaussian goes to the other Gaussian, but does not actually exchange with the 34 angstrom distance before they actually go overall into its equilibrium state. So I do realize we're a little bit short on time, so I'm not gonna mention. I'm gonna just mention very briefly one other interesting data set, which was given to me by Glenn Mühlhauser and Tufa Asafa, and here we are. And so what they're looking at now, they're looking at global process, and the global process is the cysteine exporter cytosine, which can now export cysteine out of the liposome via pH. So it's regulated by the pH itself. And for that reason, they actually attached 2 paramagnetic labels. So they have 2 sets of two mutants. In which case, one time they look at the luminal site shown in red, and one time the exponent site shown in blue. And when the pH changes, you will see that there's a conformational change. Now, this is represented in their paper previously. And you can see that very nicely in this case. Again, we have the cysteine exporter. We have the transmembrane region, which is open towards a luminal site. And then when the pH changes, it will open up to the proton, to the cytosol side, and releases the cysteine. And Stufa and Glenn, they have done that very nicely, where you can actually look at the pH change. So you have the dipolar time traces recorded as a function of pH for those two sets of mutants. So let's look at first the cytosol side. Again, we have a very broad distance distribution given, but as the pH changes from high to low, you can see that it approaches more and more shorter distance distribution. And I think that was all done with the analysis. And the same is true for luminal ends. As the pH changes from pH 9 to 5, you're approaching a shorter distance. That simply is because it goes from an open state to a closed state with a pH. Now, during the process, me and Glenn were talking severely, and we wanted to see if we can actually do the next step in global fitting. And during the previous fitting, all what was done is to look at each mutant set separately. So there was nothing done much in a global fitting fashion. There was no PK extracted, but then me and Glenn got started, and we're like, well, what if we actually take all the mutants, what is known, so in this case, it's two sets of mutants, and we also look at the pH changes, right? So we are looking at the open-closed conformation changes, but we're also going to look at the different sites. So all of them should have the same pKa. And indeed, we're able to fit it, as you can see, very relatively well, based on the residuals. And we extracted the pKa of actually pH 7. A pKa of 7 now, which is kind of different from what we expected, because the regulating residue is an aspartate 346, which should have a pKa of 4.6, but it can be pretty much reasoned that that one is probably very dabbled inside the protein, so therefore the pKa probably changes as also inside the membrane. So I know this was a lot of information, and I'm deeply sorry. I should have probably condensed it down a little bit better, but really what I wanted to show, what I really wanted to convince you all here, is that if you still have data lying around, and we all have done analysis, maybe get a fresh look at it and try out a global analysis where you're looking at the subsequent changes in either conformation, kinetics, structure, or even if you're more into like relaxation studies, look at relaxation. All those information can be globally fit and gain you a lot of information, because you can restrain that. You can fight against noise by global fit, and you gain information out of it. Of course, that all needs to be taken very, very well with care, because we can overfit model. And just because a model fit, it does not mean it's the only model that can fit. So I really want to say that as a site of warning. At last, I want to thank everybody, my collaborators, IBM, and all folks that came to my lecture. And I hope you guys have one or two questions for me. Thank you.
All right, Thomas, thank you very much for your seminar. It was very interesting. So now the floor is open for questions from the audience. So I invite whoever has question to unmute herself or himself and ask the question to Thomas directly. You can also raise your hand. So how many data laying around you had, and how many new look did you have with global analysis?
So that's a very good question, right? Because initially, when I learned EPR and DO spectroscopy specifically, I did not know, nor did I want to know, to be honest with you, how to perform a global analysis or how to set it up. So, and back then also the only tools available were DO analysis, SVD, and obviously Eric's DD program, of which all of them could only do one single trace at a time. Of course, nowadays things have changed. But so yeah, there is a large set. I have probably a lot of data set lying around that were just analyzed with DO analysis. And like, for example, all my HIV work was pretty much just done with DO analysis and DD, and need to have another look. All of it needs to be pretty much redone to get a better fit out of it. But then again, right, we should also produce new data as well.
Exactly. Let's
So,
see.
Hey Thomas, that's Jan. I have a
hi, how are
question.
you doing?
Good, thanks. Nice talk, thanks. I had a quick question, technical question on, so you do everything with LMFit, I saw. Then does that mean you all the conversion from time domain to the distance domain, is that everything you've coded yourself, or are you using other package, Python package to encode all the dipolar
Right.
physics?
So I coded myself. However, I do admit I have followed Eric Husted's program. So Eric has outlined it greatly, and so did Gunnar, Stefan, all of them have very easy, pretty much set of equations that you can just code up easily. I did code it up myself and then, but I mostly base it on Eric's DD program. If you want to get a look at it, just download DD. It's a MATLAB program. You can just open up and you can look under the hood and everything is written out.
Right. And
That
would
being
the
said,
GeoLab
DLab
GeoLab
works great as
project help
well.
for that, because it's a Python based, and before everything was MATLAB based?
Yeah, I actually great. Yeah, you're correct. So back then when I started, DLab was not out yet. So
Right.
DLab was probably one of the best things that came out for a long time because it's actually code based rather than GUI. Now I still do DLab quite significantly for all, like I didn't mention it, but all the global fits, all the parameter fits, they need to rely that there is a strong correlation to the model-free fit. So model-free fit means SVD, Gunnar's software, Stefan's software, all of that is pretty much Tikhonov regularization. All of them, DLab, give a fit, right? But this one does not describe any mathematical model. There's no Gaussian, there's no assumption whatsoever. So
Right.
in theory, if I do a parameter fit, that parameter fit should closely reproduce, or as close as possible reproduce, what the model-free fit could do. So they should go hand in hand with each other. Now, DLab, have I played extensively with DLab's parameter fits? I have not, and I wish I probably should, but I'm sure, I think Hugo Caras is nowadays doing it, and before it was, what's his name? I forgot his name, but I'm sure they did a great job. I'm 100% certain.
Yeah.
I do use DLab also, like for just pretty much reading in data, normalizing this, just normal stuff. I hope that helps.
Cool.
Yep.
Thanks.
Yeah, no worry.
Yuji, go ahead.
Hi, Thomas. Thanks you for the nice talk. Sorry, I was a bit late. I had some audio problems. I have one very naive question. Just follow what Yan just asked. So you mentioned you use this LangFit module in Python, and have you tried other modules like SciPy Optimize? And does it give a difference in terms of how time consuming it is, and what is the performance looks like
So,
for those code?
yeah, so actually, LMFit, thank you for asking the question. That's a good point. So LMFit is actually just a different version of SciPy. So it's pretty much using SciPy Optimize under the hood.
Okay.
So if you go on in the basic code and you go through it, you will see that actually everything is relying on SciPy Optimize. So it's using everything there. Yeah, there's no difference between them. because they're relying on each other. So that's all. It's just that LMFit allows you to keep things a little bit organized. That's why I'm using it. Makes things for me easier.
Yeah.
But
and when you do this, when you do the global fitting, will it be much more time consuming compared with the other packages that has been developed?
Say it one more time, sorry.
So when you do the global fitting, will it be much more time consuming compared with the other packages, like DearLab and Dear Analysis?
I don't think it's gonna, well, depends on how you code it, right? So honestly, I am not the best coder. My code normally looks rather dirty. So I got to be honest to you that I have a hint that probably DLab is outperforming me. That being said, though, yeah, the end, it just needs to get done. I mean, if it takes, my opinion, look, if it takes you 1 hour to fit compared to 1 hour and 10 minutes, it just needs to get done, I feel.
Yeah. If that's 10 minutes differences, I don't think it will be a big problem.
That's what I feel. I mean, if you can fit it in Fortran, now you're on a different ball game. I think Fortran and C++ are still outwinning Python, so that's going to be the one game changer if people could do that. But then you don't have the flexibility, so that's an issue.
Yeah. Okay, great. Thank you.
Yeah, no way.
All right. Any other questions for Thomas? So there is a question in the chat. Marina, yeah.
Hi, Thomas. Thanks a lot for your talk.
Thank you.
I was very interesting. So I have probably also a naive question, and I don't know if I got it right. But so basically, you were showing how you through your fits you minimize residual and you get really nice results. And also the next step would be to give uncertainties still in the parameter you get.
That's right.
Now, if I got it right, you still don't have it. So, and because I think
I do.
Oh, you do? Okay.
So I just didn't talk about it. I figured time would be better invested to. You're correct. Just the value itself is absolutely rubbish. So thank you, Marina. Actually, it's a very good question. So one question Marina is asking, well, how certain am I about the fit? What are my uncertainties about it? And more importantly, what are things I need to watch out for when I fit a value? When does a global fit fail? And certainly, if I get a value, I would get uncertainties, the error in those values itself. But not just that. I know I didn't talk about it, but normally I like to give it upper and lower limits. One very, very enticing thing is if your fitted value approaches the upper or lower limit, that means your fit is no good. I feel this is very important because many people mess it up. Even though you have an error, even though you have an uncertainty, doesn't mean that the fit is good. It just might mean that it's too restrained. So an iteration, not just through, I talked about a mathematical model underlying a process, but that's not just it. It's a mathematical model, but then also I urge everyone to play around with your starting coordinates, with your parameters, to use different minimization software. Like I know I use Levitt Marquardt, but there are many others. There are Powell's, there are infinite. Have fun, but try it out. If you use 2 of them, well, 3 if you want to, and all of them come to the same conclusion, plus minus a little, you're good. You're good. If you vary the upper and lower limit and you come still to the same conclusion with around the same uncertainties, you're good. If any of that fail, please revisit your data set, revisit your mathematical software. I know we all hate the idea, but think that we might have overfitted. I have done it. I have overfit data, and I was totally convinced. I was sold. And then, well, it's good to have another eye. And one of the reviewer was like, hey, Thomas, are you sure there are 6 Gaussians? Because I don't see 6 Gaussians. And dear God, he was right. She maybe, I don't know. But anyways, it's very important to be very careful with fitting. Because the same like statistics, it's a very good way of hiding information as well as teething them out. Does that answer your question?
Yeah. So I was thinking, because in a certain sense, then the noise in the data will play a role. So if you have very, very good data, then your accuracy will be higher. If it's less, so usually you might also use bootstrapping in order to get confidence level in these kind of things.
That's correct. That's correct. Actually, that's a good point. I didn't, well, since we have some time. So actually the idea of bootstrapping, so it's actually interesting too, because in the noise, because the noise is not always perfect in the data, especially DIR, because it turns out that our instruments, they're just not that reliable as NMRs yet. And sometimes they have some issues where sometimes they forget to pulse. I had it a couple of times where actually it didn't pulse for some time, but then it picked up again. And there was just the point, 1 or 2 pulses missing. I don't know why. But it comes in the fitting. When I fitted it individual traces, then it was obvious that one trace just doesn't make sense because there was a machine error. But I didn't know that, obviously, right? But then if I do a global fit, it was corrected, but then the fit was not as good because there was one point that was just an obvious outlier. So we actually tried to develop a software, a little Python program to extract an outlier. So in your in your code, in your DIR trace, what we do, I mean, I know Gunnar does bootstrap, and that's great too. But what I normally do is I fit, let's say I have a data set, a single trace data with DIR, and I am going through it and I'm doing a jackknife procedure. So I'm removing one point at a time and I'm seeing how the fluctuation is in my fitted variables. You will be able to identify that 1 or 2 outliers which are due to machine error. And that 1 or 2 outliers, if it is because DIR, our signal is not equal distributed, if not like NMR, but DIR is normally exponential distributed. So if that one point is early in time, it's really going to mess up your entire distance distribution. So it's very scary. It's not a little bit. It messes up a lot. So
I agree.
It's very, very important to play around with data. Spend 2-3 months looking at the same trace. It's okay. Better than making a mistake.
Thanks a lot.
Yeah, sorry. Thank you.
Right. Brown Smith had a question about sensitivity and background correction in the chat, but it's not very.
Where is it? I don't see it.
You don't see it in the chat window? So he said, sadly, he has to leave, but I would have asked about sensitivity to background correction.
Oh, okay. Sorry, I see it now. Sorry, I didn't see this. I would have asked about sensitivity to background correction.
So
what?
I don't know
what that means. But well, why don't we talk about it? I mean, that's okay since we have a minute or two. So background correction. So background correction, I do admit there are better ways of doing it. I personally just do an exponential. Fine, it works. It does its job. Now, that being said, we have a pity because we are very DIR, we don't really describe the background very well. And one thing what I started doing, I'm actually using the 2+1 echo nowadays for fitting. And also actually DIR Lab is doing that as well now, which is great. Initially, we always cut off data. In the end, normally what we do is we record it, we assume there's a 2+1 echo, and we cut off the last 500 nanoseconds or so, and we toss them. We're like, okay, well, you lose one, you lose some, you gain some. However, that being said, we don't have to, actually. Actually, we can just fit it. Really, I mean, in DIR Lab, they did a nice job of explaining it that, well, it's just a repeat from the initial dipolar trace. So why don't we just fit it? Don't bother about it. And even worse, actually 500 nanosecond, even 700 nanosecond, or even 1 microsecond is not enough to get really rid of the 2+1 echo because it really depends on your initial distance. So it's very risky of assuming it's not there because then you will end up getting like artifacts. And That's very tricky. So one needs to keep that in mind. In regards to background, I don't know really what the right answer there is. There's multiple ways of looking at background. You have the volume excluded background. You have this. You also need to assume the shape of the protein. So it's very tricky to really pin that down. Right now, as far as I'm doing it, I simply just say, well, there's an exponential background. I get rid of it. Here you go. So I'm just taking the easy way out. I do admit that.
All right, good. Is there any last minute question for Thomas? Okay, if not, this talk will be uploaded on YouTube, right? So it was an extensive tutorial, so people can watch it again. So thanks everybody for tuning in, and thanks Thomas for the seminar and the discussion. And yeah, I'll see you for the next IVM in 2 weeks, if I'm not mistaken.
Yeah.
All right.
Thank you.
Bye everyone.
Bye.
Bye.